Welcome back to the center stage here at Lausanne. I am excited to introduce our next panel to our Lausanne stage. This group of innovators and scientists are going to discuss innovations in the pipeline beyond amyloid-lowering drugs, new novel innovations that are going to advance the treatment pipeline for Alzheimer's in the coming years. Here to moderate this panel is Dr. Roberta Diaz-Brinton, Director of the Center for Innovation in Brain Science, the Regents Professor of Pharmacology, Neuroscience, and Neurology at the University of Arizona College of Medicine in Tucson. Roberta, over to you.
Thank you, George. And I'm excited to be moderating this session today, and thank both you and the organizers for inviting me to moderate this session, exploring the promise of novel approaches in tackling Alzheimer's disease. We've just listened to a great discussion on opportunities to build from first-in-class treatments in Alzheimer's, which is a perfect segue to this conversation now. Some of the questions that we will be considering during this session are, what are the current hurdles in Alzheimer's research and innovation? And this was touched on a little bit earlier today, but some of those hurdles are really around developing a precision medicine approach to Alzheimer's disease. Howard Phillott spoke about combination therapies, and that is definitely going to be the way of the future. And part of that will be, what's that precision combination? Well, we are going to have to address what stage of the disease a person is in, whether very early, at risk for Alzheimer's, and potentially treat at-risk individuals like we treat at-risk individuals for cardiovascular disease to essentially prevent the development of disease. We'll also have to look at the early and mid-late and maybe later stages of the disease and really ask, where is that person specifically in the course of the disease? That then allows us to tailor a precision approach to therapy for that individual. The other aspect is really looking at how we determine early on who is at risk for Alzheimer's disease. There are multiple on-ramps to Alzheimer's disease. We know that there are over 100 risk factors for Alzheimer's disease. How can we detect those individuals at risk and intervene very early for prevention and mitigation of that risk? How can we target ApoE4? That remains beyond aging, the greatest genetic risk factor for Alzheimer's disease. And that's a very exciting area for therapeutic development, to target that ApoE4 protein. The other questions that we can think about now are, how can we enable researchers and families affected by Alzheimer's disease to be knowledge generators, to become citizen scientists? And I think this is a phenomenal opportunity in this digital age for this to be a global engagement of citizen scientists around the world. And what are some of the platforms that can enable this citizen scientist for Alzheimer's disease? One of which has already been mentioned is the Brain Guide through Us Against Alzheimer's. And that's with mybrainguide.org. There's also mindcrowd.org, a platform that allows people to, again, determine how well their cognitive function is performing. There's also information at the National Institute on Aging for clinical trials in Alzheimer's disease, where people can access the clinical trials that are ongoing in Alzheimer's disease. The Alzheimer's Drug Discovery Foundation site has recently posted a phenomenal development for the current state of therapeutic development across the world and across the globe, with over 118 clinical trials currently being performed across different phases of the clinical trial spectrum, from phase 1, phase 2, and phase 3. And in that portfolio of therapeutics that are being developed in clinical testing, are therapies for my favorite, regeneration, but neuroprotection, inflammation, misfolded proteins, mitochondria, promoting mitochondrial and metabolic function, vascular disease, synaptic activity and neurotransmitter targets, as well as the genetics and epigenetics. So it's a diverse portfolio, and that's very exciting to really address the challenge, the multifaceted challenge of Alzheimer's disease. So coming back to my favorite, regeneration of the brain, we're developing and currently in phase 2 clinical trial in a clinical trial titled Regen Brain, of the first regenerative therapeutic in Alzheimer's disease to regenerate that Alzheimer's brain, to restore brain structure, brain integrity, and brain function. So with that, I will now turn it over to introduce the panelist in this exciting session. And as each of you introduce yourself, please share with us your novel therapeutic approach, your vision for your contribution in the field, and your hope for the direction in which the field can go and opportunities to achieve that goal. I'll start with Jana Hertz first, and we'll go down in alphabetical order. Jana?
Thank you, Dr. Brent. And I thank the organizers for the invitation to sit on this panel. My grandmother lived with us when I was growing up, and she had such a great, sparkling, sarcastic, funny personality, but that was taken from us by Alzheimer's. And so this is a fight that's very near and dear to my heart, and I'm very grateful for all of you who have attended this, some for many years, and your efforts to continue the fight against Alzheimer's as well. So I work at ASI. ASI has been in this fight for a long time as well, as many of you know, and we actually have many therapeutics in development across all the different mechanisms that Dr. Brinton mentioned. But I lead the site for ASI's genetics-guided dementia discovery efforts in Cambridge, Massachusetts. And here, what I hope to share with you more details on through the course of the panel discussion, we are very focused on following what human genetics has told us about Alzheimer's disease and developing therapeutics that are informed by those hypotheses. So we target genes that are expressed in the microglia of Alzheimer's, of brains, which are sort of the immune cells in the brain. You can see one on the wall here behind me, at least a cartoon of one. And we think that these cells play a really important role in sort of regulating the brain's immune response to Alzheimer's disease pathology. So we're very excited about our efforts in this area. But we know it's going to take the community, the patients, caregivers, clinicians, academics, to really help advance the biology here, to identify biomarkers to help target these promising therapeutics to the right patients at the right time, as Dr. Brinton mentioned. So excited again to be here today and to talk with all of you in more detail. But I'll pass it on next to Saul Cato.
Thanks, Jana. Good to see you. Jana and I go not way back, but pretty far back, and I think it's a great order that you've set up the introductions, because I'm a professor of neuroscience at University of California, San Francisco, but also the co-founder and CEO of Herophilus, or 5-year-old neurotherapeutics discovery company. And our approach and our belief is that we as a field, we need to do a lot, go back in some sense to the drawing board of doing deeper and more extensive preclinical modeling of disease using a new generation of more sophisticated in vitro modeling. This is human cell derived in vitro modeling. We can now culture bits of brain tissue that have been derived from the blood of patients, and we can get down to a more precision and more detailed understanding of the biological mechanisms behind disease. But we start in some sense where with the data that Jana has mentioned, with the genetic backgrounds of patients, and we seek to build models where we can then prosecute and discover in a much more scalable manner using modern technologies like automation and robotics to discover mechanisms of disease that have really not been well appreciated and understood before pushing too hard into the clinic. And this is independent of the particular therapeutic modality. There are a lot of interesting new modalities. I think this has been appreciated to be both a molecular disease as well as a systems disease. And we need to attack and study the interplay of both of these levels of biology. So it's exciting to see on the rest of this panel, other folks pursuing both pharmacological approaches, combination approaches, as well as other interventional approaches. So it's very exciting to be here, and I'll pass it on.
Great to be here. I'm Christopher Missling. I'm president and CEO of Anavex Life Sciences. We focus on the also genetically background of the disease. We believe that Alzheimer's disease is a chronic neurological disorder, which is very complex. And we have learned from early studies using genetic background of in clinical trials that the imbalance of the homeostasis of housekeeping functions are impaired in patients with Alzheimer's disease. And what we have found, though, is that the body itself has mechanisms to compensate for these imbalances and homeostasis dysfunctions. And we have activated these body-owned defense mechanism. And what's interesting is that that this approach, which was taken from the playbook from oncology, has led to in oncology immunostimulation to very successful outcomes, is here at the test to be applied also in CNS. And what's encouraging so far is that we've noticed that this approach is not only beneficial in Alzheimer's disease in clinical trials, in early clinical trials, but also seems to be able to translate into benefit in other indications. And we know that the CNS spectrum shares many pathologies like inflammation, microglia inflammation, for example. And that's what makes it encouraging to approach this.
Excellent. Hi, I'm Brent Vaughn. I'm the CEO of Cognito Therapeutics. Cognito Therapeutics is a non-invasive application of optogenetics, and we apply optogenetics to be able to directly target abnormalities and disruptions of brain electrical activity to ultimately impact both structure and function and affect disease modification in neurodegenerative diseases, beginning with Alzheimer's. We work along the premise that we know that we can change behavior and brain function directly through electrical stimulation. We've done this over the years in Parkinson's, in epilepsy, where we have the ability to go directly in with deep brain stimulation. Obviously, not something that can be done with thousands of electrodes to prevent progression of MCI to Alzheimer's or Alzheimer's progression. But we understand that relationship. We also, over the last few decades, have understood the relationship of changing protein pathologies and the impact of the ability of changing protein pathologies, especially with A-beta in Alzheimer's, to be able to ultimately see changes in electrical activity and function. And what we have been able to understand at Cognito is a non-invasive way to directly target abnormalities and disruptions in brain electrical activity and show that we can treat these as therapeutic targets. And when that happens, we see two very exciting things. We see changes in function and behavior, but we also see changes in structure. We see changes in protein pathologies. And we've realized that in the same way that protein pathologies are druggable targets to change electrical activity and behavior across the brain, we've learned that abnormalities and disruptions in electrical activity across the brain are in themselves novel druggable targets. And we've shown they change function, but also change protein pathologies and structure. We've now done this across multiple phase 2 studies, and we've shown that our therapeutic intervention has the ability to, or at least the potential to, slow and arrest the progression of Alzheimer's disease, which is our lead indication. We were granted breakthrough designation based upon our phase 2 study earlier this year. We've shown the ability to improve memory, cognition, functional symptoms as well. And one of the things that we think is most exciting, we've shown across multiple studies the ability to slow or arrest the accelerated brain atrophy and volumetric loss that is seen with Alzheimer's progression. So we are now moving forward into our phase 3 pivotal study here, primarily in the United States, and we will be looking at disease modification to affect both function and structural changes in Alzheimer's. Thank you very much.
All right, I think I'm next. My name is Dr. Christiane Rehn. I'm an assistant professor at the Cardiovascular Research Center and the McCann Center for Brain Health at the Massachusetts General Hospital and Harvard Medical School. I'm also the leader of the program for neuroprotection and exercise. So our research is interested in finding the molecular mediators of the beneficial effects of exercise to identify them as druggable targets to treat cognitive decline in aging and Alzheimer's disease. I am also the scientific co-founder of VIII Therapeutics, which is a company who seeks to also harness these protective molecular mediators to find novel therapeutics to treat neurodegeneration or neuromuscular diseases. So our, we might be a little bit different. So our research really focuses on one of the lifestyle interventions that has been proven to be effective in preventing or ameliorating the progression of Alzheimer's disease. So namely physical activity or exercise. And the idea is we know from many clinical trials, we know from studies in mouse models that exercise has these neuroprotective effects on reducing cognitive decline, on reducing the A-beta plaque burden, a reduction in neuroinflammation, in mouse models where we can look at adult hippocampal neurogenesis, so the generation of newborn neurons in the brain. We also can show that exercise is helping you to make new neurons in the brain, and even in Alzheimer's mouse models, or slow down the loss of them. So, and my lab really focuses on finding out on a molecular level, how does it work? Finding these targets and then really boost those targets, drug those targets to give you not just the benefits of exercise, but also going beyond what exercise could possibly do to protect your brain health. And one of our recent studies actually has identified one of these very promising targets. It's a novel exercise hormone called irisin, made in skeletal muscle and in the hippocampus in exercise. And we have shown it's a crucial hormone for mediating the cognitive benefits in exercise, in aging, and in Alzheimer's disease. And also using, again, we are working with preclinical mouse models. We have shown that the administration, just elevating irisin in the bloodstream of these mice, was sufficient to protect their cognitive loss in several Alzheimer mouse models, and probably by reducing neuroinflammation in these mouse models. So this is kind of a very specific approach how we think we can harness the neuroprotective effects of exercise to treat Alzheimer's disease. And I'm also very happy to be on this panel here today. It was a very great pleasure to listen to all these exciting research directions and people coming in from all these different angles. And I'm very optimistic that by tackling this horrific disease from all sides, we can find a viable target moving forward.
Thank you, everyone, for those great introductions and calls to action that you have advanced. So, coming back to the whole issue around the immune system, Janet, do you want to lead us off a little bit on why is it important to focus in on microglia? And then, as part of our conversation with each other, really thinking about, can we learn anything about the immune system from immune oncology? What's been learned there? So, Janet, do you want to lead us off?
I'd love to, thanks. So I think it's important to understand where our interest in microglia came from. Again, we've been guided by the genetics and by the data, which pointed to them starting around maybe 10 years ago or so, if we think about when some of the first genome-wide association studies were published in Alzheimer's disease. And as many of you know in the audience, if you have a background in genetics or human biology, these studies are very simple. They just compare the genomes of people with Alzheimer's disease with matched controls and look for differences. And we're up to now very large studies, some which have included DNA from over 1 million individuals. And those have yielded about 40, 50, 60 locations in the genome that significantly impact Alzheimer's risk. And if we look under those locations in the genomes, under those peaks, we see overwhelming representation of genes that are expressed in microglia. And so this was not, it was not maybe so widely appreciated prior to these studies that microglia could play a causal role in Alzheimer's disease. But, that's the evidence that was uncovered. And because DNA has a sort of unidirectional causal effect on disease, your disease doesn't go back and rewrite your DNA in every cell of your body, we can be pretty confident that these genes, these targets, are playing a causal role to drive disease. So, there's a lot of research going on now into some of these genes. TREM2 is probably the most widely studied, but more and more groups are doing very exquisite functional studies to identify the specific mechanisms by which these other variants are sort of impacting other genes, again, microglial genes. And so the role of microglia in Alzheimer's disease is also emerging. We think they may play a role in phagocytosing and eating the amyloid that's there. This is one of the things that they do, or maybe walling off the amyloid and protecting the surrounding neurons. There are also some other emerging hypotheses that they may help to facilitate rewiring of neurons around damage from amyloid and tau. And so the role and the stage at which they're relevant is still something that's being very actively explored by ourselves and others. Certainly looking at Sol, they have technology that's helping to understand some of these roles. And again, many of the labs and our collaborative partners are very active in this area.
And so that's a great segue. Sol, do you want to riff on that?
Yeah, I think the Janet gave a great description of the sort of scientific set of deductions or inferences that have been growing over time in the last decade or plus, and it's I think there's no denying, and I think that's why you have so many folks on this panel focusing on the neuroimmune axis and the general hypothesis space of either looking at the interactions of microglia with neurons, with neuron parts, synapses, which hold our memories and give us cognitive function, with other players in the brain, including astrocytes, other cell types, and getting that appreciation of how the interplay of these cells and being able to study them. Also, I should say there's a growing appreciation that we need to do primary discovery in human systems. That animal models have their place and their importance, particularly to look at, for example, toxicity before going into real human trials. But we now have ways as a field to study, these systems in vitro systems that are human based, and so I think it's a really exciting time. The interactions of microglia with the various other parts of your brain are, I should say, we're still at a let's say an early stage of understanding, and not full understanding is not required to begin studies and interventions and modulations, but it should be stated that it is there are also potentially neutral or understood or even negative effects of your immune system, as it has been well appreciated in immunology in general. So inflammatory states can both be helpful as well as deleterious to a system. And so I think this is a really exciting time, and there are many things to study and approaches to try. We have more targets. We have an appreciation that targets can work in concert. We can read out far more. I think we'll probably touch later on biomarkers. This is going to be more and more of an important way to understand and assess disease as it progresses in a patient, maybe pre-symptomally, but also to understand and to become more effective at running clinical trials. So all of this really is, I think, works together, but this renewed attention to the disease and the mechanisms behind it, I think, is really critical. So exciting times.
Terrific. So Christopher, do you want to give us your thoughts on why the immune therapies, like with anti-amyloid beta antibodies, have had so many mixed results and primarily modest, if you will? They've had target engagement.
Really great target engagement, and I want to applaud all scientists on that spectrum. I think what we learned when we set step back a minute. And this panel here had the chance to do that. That's why we hear this great ideas, is that Alzheimer's disease is a very complex disease. It's just not only, but there is a better pathology involved. But now we learn there's tau, there's inflammation, microglia inflammation, there's calcium imbalance, there's chromatin remodeling requirement, there's autophagy, insufficiency. So it's a lot of facets of what can go wrong in the body, which could then lead to Alzheimer's pathology or to cognitive impairment in dementia. And I think it's also what we have learned recently from papers from scientists that A-beta is also involved in a protective mode sometimes. And Rudy Tanzi provided a paper on that quite recently. So it's just not very trivial to grab and say, this is the cause, we remove it, and then everything is good. And I think that's what is in the chance for all of the scientists and engaging in something novel and exploring and stepping back and looking at the whole picture in a more holistic view. And I come back to the lessons learned from oncology, where there's also was 10 years ago, even before, a realization of that targeting the cancer itself, kind of like micromanaging it, is maybe not the most astute way. It is a way, but it's not the most astute way. Maybe we can use the body-own defense mechanism, which is much more potent in its entirety. And that led to the activation of the immune system to fight the cancer. And it's almost a little bit the analogy of third world support. Don't build the houses, teach them, give them the tools to build the houses, and then they will do and repair the houses themselves down the road. And that's the same here in Alzheimer's. The thinking might be better off to go in that direction. And that's what we have been done. And we're now happy that we have now a phase 3 running, which is fully enrolled, and we'll see the outcome of that probably around the time in a year from now. But that's built on that factor that we activate the body-own defense mechanism to compensate for not only A-beta aggregation, but also tau and inflammation and these other, potential harmful disorders and cellular stress effects.
Exciting. Can't wait to learn the outcomes. Congratulations. So, Dr. Ren, what about these biomarkers? It's going to be critically important that we have indicators of target engagement. Can you talk a little bit about that in your kind of complex multi-system approach of exercise?
Yeah, thank you. That's a very important question. There has been made some progress in finding good biomarkers for, pushing disease like diagnostic earlier, either with, CSF biomarkers with p-tau assays, or now even there's some promising data on using different p-tau assays in the blood to come to an earlier diagnosis. However, the biomarkers we have are still not very good in staging the disease and monitoring progress, or what, we are a lot interested in is also monitoring, success of therapies. So I think there's still a lot of work to be done. And I think there are different areas where we have to look at. So traditionally, again, when you think about the hallmarks of Alzheimer's disease, there are the plaques, there's the tau pathology, and now there's neuroinflammation that gets more and more focused. So this is the traditional approaches where people have tried to find biomarkers, meaning, markers that are changing with disease progression that we can use. I think it's a good idea also to look a little bit, like the neuroinflammation is still very underdeveloped. We don't have any good biomarkers. I mean, there's some promising biomarkers, but we don't have one that is very well accepted in clinical trials that is really already measuring the component of neuroinflammation, or even better, I think Jenna would agree, like a biomarker that would, for example, measure the beneficial activity of microglia or the destructive, that you can even say, it's not just for microglia, but it's telling us, is this microglia is engaged in clearing plaques, or is this like an angry microglia that is like destroying synapses? So I think there's a lot of work to be done. Another biomarker that has gained a lot of attention is the neurofilament light chain, which is not especially attached to one specific disease. If the idea is that it's a part of the neuron. So if you have any diseases that compromises the health of the neuron, for example, causing neuronal death, part of this is going to be released, and we can measure that. However, again, this is unspecific for different diseases. It's not completely clear if this is only happening in cell death or also cell stress. So again, I think it's promising, but we need something more targeted, especially as our moderator has pointed out, we wanted to go to precision medicine. So having the ability to very specifically monitor different disease progression. So I think that's something that also the McCann Center at MGH is very interested in finding these novel biomarkers that you could use even in a primary care setting, similar as when you go to your doctor and they check your blood for cholesterol and tell you, are you at risk for developing heart disease? Are you at risk of developing dementia, apart from the genetic risk factors that we're already having? And I think looking at, not just at tau pathology or amyloid plaques, but looking at neuroinflammation, or I'm coming from the exercise field, also looking at the changes that exercise is causing, or the changes that coming in with unbiased approaches, what are the changes that aging is causing, and using this as a marker for monitoring brain health, I think could be a very promising approach. It's going to be a long road, but I think this is something that needs to get done to move forward in the field.
Thank you, Christiane. Brent, what's a biomarker for changing electrical activity in the brain?
Well, looking at electrical activity across the brain, right? I mean, when we look at EEGs from patients that have Alzheimer's versus non-Alzheimer's patients, you can see dramatic changes right at baseline. And one of the things that was seen early on and known throughout the literature is that when you look at Alzheimer's patients, you see a relative absence of brain activity across the gamma frequency. And throughout the literature, we know that gamma frequency oscillations are integral not only into how the brain communicates and processes information, but ultimately trigger so many of the different protein, chemokine, cytokine changes that we've now shown the relationship throughout clinical studies. I think that when we look at the ability to change the pathoelectrophysiology in the brain, we see some very dramatic changes. What we just heard Jan and Saul talk a little bit about the role of microglia in this disease. One of the first things, in fact, the seminal paper for us that was published a number of years ago by our co-founders out of MIT in Nature showed that gamma frequency stimulation activated microglia in the visual cortex and then across the brain. And we saw microglia mediated clearance of A beta and hyperphosphorylated tau. Right, so working in exactly the way that Jen was just talking about. And so I think that when you start to look at engagement, you can see target engagement right away. We can see changes in the EEG immediately upon providing gamma frequency stimulation, and we see entrainment and coherence build across the brain. We know that neurons that fire together increasingly wire together, and so in the same way that Jenna suggested that microglia might also be implicated in how the brain changes or adapts its synaptic connectivity, we see that. We see changes in fMRI showing increased connectivity through default mode network. And so I think when you think about kind of biomarkers in this space, I think that we're going to start to see a progression. We're going to see a progression from this idea of, the term was used earlier about overly precise, like immuno-oncology, from this idea of just looking for bad agents, right? It's not just about counting up A beta plaques or just looking at tau. It's going to evolve into a little bit more of a systems biology approach. And I think the thing that everyone's talking about here, which is how we approach the disease, is looking at microglia activation, see how that involves astrocytes, and start to look at chemokine and cytokine expression changes that shows that we are helping nudge the brain or react to the brain's ability to start to regain homeostasis, which it is clearly lost in the progression of Alzheimer's.
Terrific. Thank you. So moving forward into the kind of the innovations that are going to be advancing the therapeutic development space, what do you see in the future? And Dr. Missling, Christopher, do you want to kick us off on that?
Absolutely. So I think what you just mentioned before is really important, the precision medicine. Maybe one of the reasons also we have not been able to successfully see a product really a breakthrough in this pathology is that maybe the patient population were not homogeneous enough given the complexity of the disease. But I think one of the key lessons learned for the future, in my opinion, would be to educate, and we're doing this today in this panel here, to educate the audience that there is a lot going on, and it's very important to keep an open mind and keep learning and appreciating that the science is evolving to learning more and more, and using precision medicine to select better patient populations or more appropriate patient population for treatment, and also finding biomarkers which truly respond to the therapy. So not only reducing A beta with the antibodies do very nicely, but the correlation with the outcome is extremely important, because that's ultimately is the proof of that the biomarker is resilient and supportive of the treatment. So this would be my encouragement, to be open-minded, to be aware that more is coming. And this group here really describes that very nicely.
Christiane, do you want to take off on that? What do you see as the up and coming?
Of course, since our lab is very interested in the neuroprotective effects of exercise, I think that's an untapped resource for finding novel targets. So we basically have this reverse engineering approach. So we are looking at how the body itself can help to build resilience against developing Alzheimer's disease or slow down the progression. So as we talked about, instead of just going onto one specific pathology, we ask the body to tell us what is working and what is not working, and then we are trying to do the reverse engineering to then translate these good effects into molecular targets. So, and that's an approach that hasn't really been tapped into, probably because exercise is similar to Alzheimer's disease. It's a multi-system crosstalk, complicated process going on. So it took some time to dissect out what are important players, what are the right molecular mediators. Of course, we are optimistic about our target, but I think there is still much more to learn by looking into how, either the hippocampus is responding to exercise. There's a lot of interest in the, different peripheral organs, such as the skeletal muscle crosstalk. So seeing how, exercising your muscles is releasing hormones that actually can provide this neuroprotection in the brain. It looks like part of that is going through the neuroinflammation angle that my previous speakers, but we haven't really, understood the whole systemic process yet. So I'm very optimistic, especially with these new, higher throughput single cell sequencing technologies, that we can basically untangle this, complicated process and boil it down to the molecular message that we need to get.
Thank you. So I'd like to switch gears a little bit now and move from kind of thinking specifically and more broadly. A common theme that each of us share is moving discovery that has been made at the bench through clinical development to commercialization. On my end, we've developed a startup, New Therapeutics, it's NEU Therapeutics, to advance commercialization of our regenerative therapeutic, because if we don't commercialize, we don't ultimately reach our target, which is the Alzheimer's patients. So I'd like for each of you to kind of share some of the challenges and successes and lessons learned for your journey on moving from discovery to ultimately to commercialization. And to kick us off, Saul, you want to kick us off on that?
I'm probably the furthest in this group from commercialization, so I won't take up too much time. I'll pass it off. I will say that I think everybody would, a hazard that would agree that getting through clinical trials with a higher degree of success probability and a greater degree of confidence is critical, especially given the track record that we've had in the field. And in order to do that, we've got to get smarter about readouts and about patient stratification going in. And I think all of the folks here also would appreciate that, starting with genetics, following up with measurable phenotypes and biomarkers, but But this is how we get through clinical trials with wins rather than sort of losses or neutral results.
Perfect. Jenna.
Yeah, I mean, so ASI has been doing this already, doing this for a long time, going back to Aricept, right, bringing therapeutics to market for Alzheimer's disease. But I completely agree with what Saul said. There are certainly ways that we are trying to do that in a more efficient way. Targets that have been identified by human genetics have shown that they are twice as likely to succeed in clinical development to get to the point of approval. And that's across multiple therapeutic areas, been shown by multiple groups at this point. So that's one of the reasons that this center was established and that we're continuing to hunt those genetic targets. I think that, again, not to tie this back to biomarkers again, one of my favorite topics, but I think that, we've seen that clinical trials could be faster if we are able to use more dynamic biomarker endpoints, ultimately potentially as a proxy for cognitive or functional endpoints. And so I think that obviously the community is very active in that area across multiple companies and academic groups. And we would say that we want to identify biomarkers that are relevant not just to capturing disease overall, but to specific pathophysiology pathways that are involved in disease so that we can specifically read out the pathways that we're targeting.
So, kind of to expand on that a little bit, Brent, can you talk a little bit about these surrogate biomarkers, right? So gamma oscillations, are a great biomarker for multiple systems of biology in the brain. Are you looking at that as a surrogate biomarker?
Yeah, so historically the bar to be able to achieve the surrogate label at the FDA has been inordinately high. I think it does not even take two hands worth of fingers to count the recognized surrogate biomarkers at the FDA. And so I think surrogate is probably a longer and higher mission than ultimately trying to figure out how to get your novel therapeutic intervention across the finish line. We certainly look at changes in the electrophysiology across the brain to show target engagement. Right. So when we provide a gamma frequency stimulation, we can see entrainment and we can see coherence building across the brain. And so we use that as a biomarker of target engagement. It's a bit of a checkpoint for us to tell us whether or not, when you think about some of the heterogeneity that we see in these patient populations that Saul was talking about, this is not a one size fits all when you think about addressing the etiology of disease progression. So I think that that's interesting. I think when we're looking for what could ultimately become surrogate biomarkers, we're quite excited about looking at structural changes that we think correlate with the functional changes we like to see in these patients. So as we all know, there's been a very large focus on the ability to change concentrations of A beta and to remove A beta plaque. And that has been effectively used with a conditional approval, I would say darn close to a surrogate in Alzheimer's disease quite recently. And I think that whether or not you believe in that and you think that that is the right modality or the right surrogate to be or in biomarker to be thinking about in this disease, no one can argue that it has increased the acceleration of innovation and the path of products coming to market, right? There are antibodies quickly moving down the path from Lilly, from Eisai, who's represented here. There's a number of different that are going to be moving down that path. And ultimately, I think most of us probably believe, even if you're not a huge fan of a particular mechanism, that bringing increased opportunity and innovation closer to physicians and patients is ultimately a tide that lifts all boats. And so I think for us, when we think about biomarkers, we're trying, besides looking at maybe entrainment and coherence across changes in brain activity, we're also quite interested in looking at some of the things like volumetric change, looking at the ability to preserve hippocampal volume, whole brain volume, like we've seen in clinical studies. And so I think that there's some ripe areas there. But to your original point, looking at changes in electrophysiology is quite powerful. electricity is the fundamental currency of the brain, right? Our brains have evolved to generate, transmit, and store electrical activity. And so I think as we start to understand how to interrogate this more and more, we're going to start to see things that are more than just target engagement that hopefully will become, if not surrogates, I think we should at least be aiming for biomarkers that are predictive of on mechanism and efficacious effect.
Terrific.
I can add something to that. I totally agree with that. EGERP was one of the fundamental inclusion criteria or part of the phase 2A trial in Alzheimer's disease with our drug Anavex 273 before moving into the large stage phase 3 study. And we did see a very nice signal of P300, improving compared to patients who did not get enough exposure. So that was an instrumental part of, moving forward with this. So this is one thing. The other thing regarding commercialization, we're not there yet, but we hope to get there. But what I would say, what we have done consistently in all our clinical trials, we have engaged with patients advocacy, with the caretakers, and with the investigators to educate them and also ask them and work with them together before we finalize the clinical trial protocol in order to get their buy-in going forward. And it's really, I think it's the best you can do at this stage before you are in a commercialization level, because it starts very early, the interaction. Because what we try to do is find a solution for the patients. So the patients are, for us, in the front and center of all of this.
Perfect. Thank you. So there's a couple of questions from the listeners, and I'm going to open this up to whoever wants to jump in here. So what do you think there's a merit in targeting the microbiome?
I would say yes. And the reason why I say that, because we already have preliminary data on that as well. So we did test biomicrobiota of our drug in the phase 2A study. And we have noticed that when you look at the papers, at the science, that Alzheimer's patients have on average a lower abundance and variety of biomicrobiota. And what we noticed with the treatment, there was a improvement of the balance towards more healthy volunteer levels. So that could indicate that there is indeed a blood-brain and gut microbiota axis which are communicating. And I think that's what we always know, have a gut feeling. And when you're stressed out, you have gut issues. So that goes without saying that that precedes, even evolution. So the only question is, what comes first? Is there a correlation of treating the gut microbiota and benefiting in a CNS way? Or is it just a way of looking at it as a biomarker of response? And that's what we are finding out soon, because we now included in our late stage trial in the Parkinson's disease dementia trial, the gut microbiota
Oh, great.
Lost in space.
Okay, so, but coming back to some of these other issues, we're almost at the 10-minute mark when we'll be closing out this session. So I wanted to really move us to asking the question, what are the barriers? What are the opportunities? What are the strategies that are going to be critical to advancing each of our innovations globally? Alzheimer's is a global disease. It's a global challenge. How can we impact the global resources to advance rapid and safe development of novel therapeutics. And I'm just going to throw out there, one of the challenges is the multifaceted regulatory ecosystem where, with good reason, there are many very different requirements across many different nations. Gratefully, the Eurozone is more homogeneous now. But talk to us about, and I'll start out with you, Jenna. Talk to us about this. You probably have the most experience with this international global regulatory system. Is this an area where improvements, efficiencies could really advance therapeutic development and reaching this global population of Alzheimer's?
Yeah, that's a great question. And being at the discovery end of the process, I'm not sure I'm very well placed to comment on the regulatory end towards the end of development. But I think the topic of how do we work globally is critical actually for our discovery efforts. And if you think about the types of genetics research I've been talking about, globally, Alzheimer's genetics has been conducted primarily in white European populations. And so we really need to improve the diversity of data sets that are used for research. And that includes, genetic data sets, but also biomarker data sets. I think we're starting to see more efforts in this area, which I'm really excited about. But we have every expectation that by being more global and more diverse in our research approaches, we're going to identify new targets, novel biology, novel biomarkers. And I'm sure things like exercise and microbiome are going to be very different globally. And we'll surely learn new insights from those types of data as well.
Saul, you want to weigh in on a global, advancement?
Well, no, I think Jenna put her finger on it. Starting for the much broader base, which we now have genetic information across the world in many different areas and from many different countries is fantastic. We're really discovering that this is a collection of different etiologies that are producing this disease category. But you kind of touched on the opportunity to leverage the global stage for clinical trials, and there are some interesting opportunities now to do clinical trials out of the U.S. And out of even Western Europe, and I think that's emerging, and it could be a way to be more efficient. I think we do need to figure out ways to have more entrants in what is a costly, expensive, and difficult thing to do, which is run an Alzheimer's trial. And this may be a way forward for companies like that, which may have very very exciting science and very exciting approaches, but may not have the ability to launch a large trial at first. So good
points. Christiane, your thoughts on the global challenge?
I think it's something that is not just a unique challenge to Alzheimer's disease, but just if you think about how drug development and then eventually drug participation in drug use is then later on regulated. If you just think about the discussion we have now around the COVID vaccines as one of these examples. So I think it's something where regulatorily we have to work together and have some vision. How do we deal with the situation that a lot of the drug development and discovery is being done in the rich countries of the world by manufacturers who, with good reason, also have an interest in having profits. But on the other hand, how do we then come up with strategies if and when we develop a therapeutic, then also have countries that have less resources than we have being able to participate in the goods. And I know that also for HIV drugs, there was a lot of discussions how we are doing that. I don't think we have found a perfect solution. And I'm not an economist. I cannot tell you how exactly you have to do that it's fair for everyone. But I think especially when we're thinking about that these therapies we are developing, the more precision they are, the more they're geared to maybe immune therapies. So there are costly therapies, and we have to find a way to make this then, that there's equity and participation of everyone if you want to solve this problem globally.
Yeah, Brent?
Yeah, I think that obviously, a lot of the key points have been nailed by some of the ones that have gone before me already. Being able to think about engaging other populations in your clinical trial process earlier. We are gearing up. We will be starting our phase 3 pivotal trial, which will be the basis for our submission and approval. We'll start that in Q1. We're already in discussions across Asia to add cohorts in some of the countries there that would allow us to actually look at pivotal cohorts in Asia that would combine with our US data, but that would run in parallel. So starting to move away from the more sequential idea of US market first and then spending years starting to knock over the other dominoes as you move across the world. I think number two, some of these therapeutic modalities just fit better into different economies and different markets and different healthcare distribution systems than others. We're lucky in that we have a non-invasive therapy that through our clinical studies, although it is prescribed and the patient is first shown how to interact with the therapeutic platform in the physician's office. After that, it is a home-based treatment. Patients do this at home with their care partners and have shown an incredibly high degree of adherence and compliance to do this on a daily basis. And so I think for us, some of the international markets make a lot of sense because it is easier, especially in areas like for China, for example, where half the population lives in a relatively rural setting, the ability to come into a tier 1 or a tier 2 city to be able to get prescribed your treatment, but then to be able to return home, to be able and still participate in the treatment. I think that lends itself well to being able to move outside of a more US centric market or even US Western Europe centric approach. But these things can be quite hard. And I think it's really case by case as you start to look at the intersection between the treatment modality and the different countries that you're trying to move into.
So I think that's a great segue. The ability to move therapy from a centralized kind of depository to now a home base approach is really going to be very, very important and will be a game changer. Obviously, the digital technologies like those that are being currently developed for monitoring one's own cognitive function is a great step in that direction. So with now that we're rapidly coming to a close, I want to thank you all for this fantastic discussion today, your thoughts, your insights and perspectives as we continue to disrupt and innovate the Alzheimer's field. I would like to thank the Lausanne VIII organizers and George again for inviting us to be part of this year's events. I only hope that we can be together soon and we'll be reporting on great new discoveries and cures in the near future. And with that, I would like to turn it back to George. Thank you again.
Thank you, Roberta. Thank you for your animated moderation of that fascinating panel, going back into the science to demonstrate that there is much, much more to come, both in clinical trials, but even before clinical trials and the deeper scientific understanding of the disease.